Okay. So we're very pleased to have Dr. Anat Perry with us today. Anat completed her PhD at the Hebrew University under the supervision of the late Professor Shlomo Benton of blessed memory. In her PhD, she focused on brain mechanisms that enable the understanding of others. I know Anat from then, and I recall at the time how proud Shlomo was of Anat and how excited he was about her findings with mu rhythms. She then continued to do her first postdoc with Professor Shimon Shammai Tzuri at Haifa University, and then another postdoc with Professor Robert Knight at the Helen Wills Neuroscience Institute at UC Berkeley. She's now faculty at the psychology department at the Hebrew University of Jerusalem and the head of the social cognitive neuroscience lab. She investigates the neural mechanisms that underlie empathy and also looking into interpersonal space and implicit social behavior. We're very glad to have you, Anat, with us in our seminar, so welcome.
Thank you, Sharon, and everyone for being here. I'm really honored. I was really happy when Sharon invited me to come. I think even last year.
Yeah, I think 3 years. I mean, this is the 3rd year, and you finally accepted my invitation.
Yeah, I was on maternity leave, and then we had COVID-19, and then we said, you know, this is the way that we have to do it anyway, and we won't postpone it anymore. And I'm so happy with this initiative. I keep following Sharon's, emails and mailing list and seeing this wonderful group and wonderful talks. And I think if we do have to have these talks online, then at least we take advantages of, this international opportunity. So I'm glad to be here. Let me share my screen with all of you so that now you all see my slides, right? My presentation. So I won't be able to see you too much, but I hope that I'll be able to follow. So I wanted to talk to you today about the contribution of different information channels to different facets of empathy. And like Sharon said, I'm a PI of the Social Cognitive Neuroscience Lab at the Hebrew University. And most days my lab looks something like this. So we take opportunity of cognitive neuroscience tools, mostly EEG, some physiological measures. We want to start looking into eye tracker methods, dual EEG, and of course behavioral methods. And what we study is questions that relate to social interactions and mainly to empathy. I told Sharon I hope it would be interesting for a vision crowd, but you'll see that I'll also in some ways talk about vision, maybe differently from what cognitive neuroscience usually think of it, but I hope it will still be interesting for you, and it will definitely be interesting for me to hear your take about this work. And this is how our lab work looks right now. And so it's very, very different. And it also, I think we all know it affects all of our work. It also affected some of the things that I'm going to show you. So a lot of the work is the work in progress, especially the neuroscience aspects of it. So I'll show you what we plan to do or what we've started doing. But then we shifted to do some things that we can do online, and I'll show you more behavioral work than we planned to do. And I think it's still very interesting. I hope you'll find it interesting too. So we'll start with an introduction of what empathy is and some evidence for experience sharing and mentalizing networks in the brain. It will be a kind of long introduction so that I can show you some of the work that I've done during my postdoc at UC Berkeley, but I don't want it to be too long. So I might skip at some point some of this work and continue to what I really want to focus on, which is the recent work that we're doing in the past few years. Which is really going into more naturalistic tasks for studying empathy. This was the main focus since I arrived at the Hebrew University. And the talk today will focus on contribution of different information channels, mostly auditory versus visual channels, on how we understand other people. And the last question, and that's, I think, why it's in some ways relevant to vision science as well, is why do we feel that seeing each other is so important, especially in these interpersonal talks, when you share emotional information, when you empathize with others? Why do we feel we really need to see the other person? What does what does that contribute to the interaction? And this is really ongoing work that we just started, and I'd really like to, you're the first one seeing these results, and I'd really like to hear your thoughts about them. So my plan is to get to that. So I might skip other parts if I see that we don't get there on time. So I like to start my talks with asking students or the crowd, what empathy is. Does someone want to take a shot?
So I want to ask people you can unmute yourself for this for this part, and sympathizing with others.
Okay. What's sympathizing though, right? That's also using another vague term.
Mirroring, trying to mirror the feelings of the others.
So when we ask, so it's a good thing to think about, right? Where a lot of these social terms are, when we start thinking about them, they're very vague. And when we ask, a class of students to come up and even write down their definitions, I get things from mirroring, simulating the emotions of other people, sharing their experiences, feeling what the other person is feeling, some things in the line of what Sharon just said. Other people might say that empathy is taking the perspective of someone else, trying to understand what other people are feeling, putting yourself into their shoes, trying to understand what I would feel if I would be in the same situation. And note that these are very different things. And indeed, people have and I like to, give you another example from Edgar Allan Poe, which says, when I wish to find out, now I can't see it myself, how wise or how stupid or how good or how wicked is anyone, or what are his thoughts at the moment, I fashion the expression of my face as accurately as possible in accordance with the expression of his, and then wait to see what thoughts or sentiments arise in my mind or heart, as if to match or correspond with the expression. So when we try to think about what empathy is, or what does it mean to understand other people, people have gave many different definitions, and I want to put most of them into these two broad definitions today. One I'll call experience sharing, or affective or emotional empathy. And this relates to things like emotional contagion, although that's also something that you might see in animals or in babies. And in experience sharing, in the terms that we use in empathy, we want this emotional contagion, but also with the sense that we know that we are feeling the emotion of someone else, right? And not just feeling it in ourselves. It's related to things like Sharon said, like simulating the emotions of others, feeling what the other is feeling. And as I said, another synonym of this term would be affective or emotional empathy. And other people talk about empathy in a more cognitive sense, which we'll also call mentalizing or cognitive empathy. And that's related to concepts like theory of mind and understanding that other people have intentions, emotions, beliefs that are different from our own. They have knowledge that's different from our own, and they also have emotions, an emotional experience that's different from our own. And we'll see during our talk today that people have studied them for a long time in very separate labs and separate streams, and came to separate conclusions. And when we get to my talk, to the next part of my talk, I want to start thinking how we can study these together. And of course, in real life, they usually happen together, right? We talk to a friend, we try to take their perspective, we understand their point of view, but we also feel their emotions and feel with them. So I'll start with this notion of experience sharing, which, like Sharon said in her introduction of my work, is what I've been studying for a long time. And the intuition is something like that, right? We know that when we see other people, the yawning effect is something that we're all familiar with. I'll move through this quickly because it's noon. And when we see other people yawning, a lot of times we find ourselves yawning as well, right? This is known not just in animals, but not just in humans, but also in other animals, but mostly in social animals that live together. So this probably has some social effect of why we imitate automatically these kinds of behaviors. And we know this is true not just for yawning, but also for different emotions. When we see other people smiling, we sometimes can't help ourselves and smile as well, and even feel some kind of happiness or joy. This happens more when we look at young children or baby, more when we know these people, and so forth. and things that are often used. I see just Sharon, which is perfect. I don't know the reactions of others, but this is classic when we have a crowd of people to see and hear and, you know, feel the emotions of the crowd. These kinds of stimuli are often used in empathy labs. It's called empathy for pain paradigms because they work so well, right? You actually have this visceral feeling in your body, right? You feel what the other is feeling, and that's part of what empathy is. So these are very common in empathy research, mine and from others as well. And when we think about the biological feasibility of such a system, then the mirror neuron system found in monkeys in the early 90s already was something that was really thought of as, you know, a perfect system to match, to be responsible, to enable this kind of simulation, right? A system that's active when the monkey does specific motor actions, goal-directed actions, but also when the monkey sees someone else, monkey or human, do similar actions. And since the late 90s until today, there's lots of evidence for human mirror system. Usually most research is not focused on humans, on neurons, on specific neurons, but networks, mechanisms that enable such kind of simulations. I think there is still a very strong debate on what this network or regions are good for, right? What do they do? How much they actually contribute to empathy or to imitation or to learning, but they're clearly there. We have evidence for such a network from fMRI research, mostly focused on this network of frontal, parietal, and sensory motor regions. There's evidence for EEG. Some of it is research that I've done during my PhD with Shlomo Bentin, focused around the sensory motor cortex on electrodes C3 and C4, which you can see here, which are over the sensory motor cortex. And show this kind of suppression, which is a proxy for neuronal activation in these regions to doing different actions and seeing someone else perform the same actions, but also to things that are more than actions, seeing facial expressions, trying to understand others' emotions. And there's evidence from lesion patients, from fMRI, from EEG, that this is not only related to motor simulation, but also to, like we just saw, experiencing pain. So when you experience pain yourself, or when you see someone else experience pain, you see a whole network of regions that some of these regions, right, not all of them, but to some extent, this pain network, especially the ACC, the insula, and part of the sensory motor cortices are active for both seeing someone in pain and feeling pain yourself, and similarly for other emotions as well. We have evidence from lesion patients in these regions that also show some deficits in understanding the emotions of others. And finally, we have evidence from both single unit recordings and electrocorticography, or ECOG. And I can show you an example of this from kind of recent work that I've done through my postdoc. And when I did this postdoc at UC Berkeley, I really wanted to, my main interest was already in these empathy networks and social questions, but we said, let's start with the simple questions of whether we can find these mirror neurons or mirror regions in ECOG as well, and what we can learn from that. And of course, if any of you have experience with these kinds of experiments or with any patient work, this takes a long time. So it took 3 to 4 years to just collect ECOG data from different hospitals around the US. And we never got to the more social aspects, which we're now running in the lab with different tools. I'm debating whether to skip this. I'll just give you this as a background to understand the systems that we work in. So EEG mu suppression is, like I said, these electrodes over the sensory motor cortex that measure, what we measure is mu suppression in 8 to 13 hertz range that are suppressed when one performs or views a goal-directed action. And it's thought to be a proxy of mirroring or simulation. And the classic example, this is not from my work, but from Lindsay Oberman already in 2005, is that when we tell participants to move their hand, we see the suppression. Here, you can see my mouse, right? You can see suppression in these different electrodes, but we also see, although to a smaller degree, this kind of suppression when we see someone else's hand moving, but not when we see non-biological motion. So for example, a ball moving on the screen. And interestingly, what some researchers have found is that, for example, people with autism spectrum disorders show regular immunosuppression when they move their hand, but not when they see someone else's hands moving. Now, I have to say this has been replicated by some groups, but not by others. So with autism spectrum disorders, I think there's still a lot of questions how relevant this system really is, or how much, or is there really a deficit in ASD populations? In all of them, or maybe in some of them and so forth. The groups are mostly small, but in controls, this is something that we see again and again. And an example from some of our work to the more social aspects, we also see this for facial expression. So for example, this is Noga Ansenberg work with me and Hilal Abiezer from a few years back, where we showed people facial expressions. So this is an example of a facial expression that you would see. Or something like this. Note, this is exactly the same expression. It's actually the same timing and everything, but from a different angle, from a different camera. And we showed people these kinds of videos from different emotions, and we asked them different questions so they could recognize the emotion, and we see that they recognize them to the same degree, whether they're turned towards them or a bit away. They rate their intensity as the same level. But when we ask different kinds of questions about relevance, how much did you feel it was relevant to you or expressed towards you or important for you to notice, then you see this big difference, right? If you see someone doing this facial expression, let's say of disgust towards you, you either think there's something wrong with you or something wrong that you should notice, right? That's in your back or something like that. So you get this difference here, and you also get this similar difference in immunosuppression. So these electrodes really notice something that's related to understanding the expressions of others, the intentions of others, important for biological motion, and so forth. This is some ECOG evidence. I'm wondering, I'll give you just a taste of it. So this is the work that I told you I did in UC Berkeley a few years back. ECOG is work with patients. I don't think I have to give an introduction. I'm hoping most of you are familiar with what ECOG is, but we had 10 patients who had coverage on these relevant regions of parietal, frontal, or sensory motor regions. Some of them had coverage in all of these or two of these regions. And we could look at high gamma activity, which we know is related both to single unit activity, to bold signal and fMRI, and really bridges the gap between these fMRI results and EEG results in humans, and also between human and animal work. And importantly, we can look at single trials and really see what happens trial by trial. And like I said, I wanted to ask really social questions, but we started with basic imitation. And to look at this basic mirroring paradigm, participants heard these were patients in the hospital, they heard a beep, and they knew that after this beep sound, they would see a short clip of a grasping action towards either a cup, a bottle, or a pencil. These objects were also laid in front of them on a table near their hospital bed. Then they had a waiting period, which changed between 2 to 2.5 seconds, and heard another beep, after which they had to do the same grasping action themselves to imitate the same thing. And what you see here is an example of one subject, just to see the kind of information that we can get. So this is a sanity check of an electrode from the auditory cortex, not related to the mirroring sites we wanted to measure, but just to give you an indication of how to look at this kind of data. What we see here on the x-axis is exactly the same timeline that you see here: the beep, the viewing, the rest, and the grasp. What you see on the y-axis here is single trials. So every line here is, every row is a trial in the experiment, and the colors indicate activation, right? This high gamma signal. So you can clearly see in red that the high gamma signal in the auditory cortex is really, is clearly for the beep sound and noisy everywhere else, right? Another example is looking at these grasping electrodes. Now, you're not supposed to see this at exactly the same time because the participants differed trial by trial at when exactly they did the grasping towards the specific object. But you can see in each and every trial here, you can see this high gamma signal at the moment of the grasp, when they grasped the specific object. You can also see something here for when they started moving their arm, right? But you can clearly see it from the grasp. This is a clear motor electrode. But what we're interested in is these kinds of electrodes, which we call the mirror electrodes, that were active both for the grasping in a similar manner, but also for the viewing. And if it's unclear here on the single trial level, I'll show you how it looks on average. This is the exact same electrode. So we see on average that there's heightened activation for viewing, then nothing during rest, and then it goes up again during the grasping. So we noticed these mirror sites in sensory motor, parietal, and frontal cortices, and we could look at different things that influence them or how they act differently. I won't go into the details. I'll show you one example. We showed that there are different kinds of electrodes. Between the electrodes we measured, we found two different kinds of activation, one exactly like I showed you now, which we called pure mirroring. They were active just for the viewing and then for the grasping. And others, which we named mnemonic sites, and I think are really, related more to working memory or rehearsing the action, which were active during viewing. But when we looked at what happens in between, they were also active in between. So in some ways, they were rehearsing what they later had to do and then performing the action. And this is, of course, not something that we still have to look into, but we did differentiate these two kinds of electrodes. We also looked into differences between timing of the different sites and so forth, but I won't go into this now. The last thing I wanted to show you that we did look at is to look at the relationship between the high gamma activation and lower frequencies, which we can all measure it with EEG or with MEG in the lab. And that was important for us for some sanity check of the work that we usually do in the lab. How is it really related to what's happening in the brain? And we can see here nicely now on the y-axis, we have frequencies. And we can see that during the same viewing and grasping period, when we have high gamma activation, we also see the suppression in alpha and in beta rhythms. And we can also see it in some of the sites trial by trial. And for those of you that are familiar with EEG or fMRI work, this is really impossible, usually in most paradigms, to see something so clear trial by trial. But here you could really see this suppression, even in the lower frequencies in each and every trial. And this was clearer over the sensory motor cortex, where we actually look in EEG to see this mu suppression. So you can see it. If for high gamma, we see activation in frontal and parietal electrodes, you see it much less clearly for this alpha and beta suppression. So really, it gives us some reassurance that what we're measuring in the EEG, we see some of the signal, not all of it, but at least it corresponds with what happens in high gamma in the brain. So all this was to say that there's a lot of evidence looking at different networks that are involved in experience sharing, from imitation and goal-directed actions to experiencing pain, understanding facial expressions, and so forth. And the focus has been on these mirroring networks and regions and on the pain matrix network. But at the same time, for at least 2 decades, people have been also studying what they called empathy, but it was a very different kind of empathy, right? They were focused on mentalizing and theory of mind and more cognitive aspects of empathy. And I'll just give you I think, 1 or 2 slides on how this was measured. So examples of these, and this is probably familiar to those of you who have background in developmental psychology, is looking at these theory of mind tasks that are used on kids, right? So for example, this Sally Ann task is a task where, this is Sally. Sally has a basket. This is Ann. Ann has a box. Sally has a marble. She puts the marble in her basket and then she leaves. And Ann moves the marble, right? And then Sally comes back and you're asked, where will Sally look? And if you're a kid around the age of 3 or 4, you understand that you know something that Sally doesn't know. But kids at a younger age, or kids on the autism spectrum might understand this only at a much older age. For adults in fMRI experiments, for example, you use these kinds of stories, these kinds of vignettes. So this is an example of a false belief sample story. John told Emily that he had a Porsche. Actually, his car is a Ford. Emily doesn't know anything about cars though, so she believed John. When Emily sees John's car, she thinks it is a Porsche or Ford. And adults do this well. So you're at a ceiling effect if you're attentive to the task, but at least researchers can look and see what happens in the brain when you do this kind of task versus when you do a lot of very nice controls for this task, which ask other questions that relate to people, but not to thinking about what they think about, or relate to objects, or relate to, lots of different controls, really nice controls that are used. A lot of this work is work by Rebecca Sachs and others that was really controlled and nicely done. And the regions that they found were, not surprisingly, maybe very different regions, right? Because the tasks are very, very different tasks. So 2 main regions are these ventromedial prefrontal cortex and the TPJ, the temporal parietal junction. But what happens in real life? And I think in social psychology in general and in social neuroscience specifically, there's been this movement in the last few years that in order to understand what happens in real life, we have to switch, although I think switch is not the right word, but also use very much more naturalistic paradigms. And I think really the best solution for, at least in my opinion, and what we do in my lab, is to really use both, to try to use both controlled and naturalistic stimuli at the same time. So in real life, you don't see these short vignettes, you don't see still pictures of people in pain, or just their hands not connected to their body, or just their faces, or they're not black and white, and so forth. And this is where I wanted to really share our recent work with you. So when I joined should I stop for questions, Sharon?
As you feel. I mean,
if there are any questions on the intro, feel free to interrupt.
Actually, can I ask a question, please?
Yes, sure.
I was wondering if orientation of view matters. So is there some sort of normalization? If you in one orientation and then you imitate in a different direction or orientation, is it normalized somehow?
What do you mean by normalized?
So is it view independent or orientation independent?
So I don't think that most of the research done on that, it's an excellent question. I think that most research was not really controlled. Some of the tasks used a hand that's really towards you, like you were watching someone else. Some of these tasks used hands that were like you were seeing your own hand in your own direction. And results are more or less the same, but I don't know if a study that really compared the two to see what happens when you see it in your orientation versus in someone else's orientation. I think there's been some work by Sylvie Frankel-Toledo, who's now in Ariel University, that focused on lesion patients and how how easy or difficult it is for them when they see a hand in their orientation or the other way around. But in terms of neuroscience, I think it's actually mixed. I don't think someone actually compared the two. It's a good question.
Interesting. Thank you.
So I'll go on to what we're doing now, and this is things that are still happening. So I'm happy to hear any feedback, suggestions, comments on this work. And so when I joined the Hebrew University in 2017, I knew what I wanted to do is really create these much more naturalistic tasks that I think can measure, first relate more to what happens in real life, but also can measure both these mentalizing aspects and these experience sharing aspects together. And what we did is we created what we call the Israeli Empathic Accuracy Stimuli Set. We're not the only ones who did these kinds of tasks, although it's modified in different ways. So I did this in collaboration with Jamil Zaki, who also has kind of different, but a similar American version of this kind of task in the US. And there are some British researchers who did something similar there. But the idea is that we brought people to the lab, participants, some were students, some were people from the community. Note, these are not actors. And we took videos of them telling real autobiographical, emotional stories from their lives, okay? They had to tell these 2-3 minute short stories that really happened to them. And we filmed them telling these stories. We also had, what you don't see here is we had physiological measures taken from them while they're telling the story. So they have electrodes here below, under their clothes, measuring heart rate and galvanic skin response. But we asked them to just tell their stories as naturally as possible. They knew they were going to be recorded. So these are not very private stories, but emotional stories that they were willing to share with other people. And let me see if I'll try to show you this example. I hope it works on Zoom.
איך שהרגשתי בו, הייתי בכיתה ג' אני חושבת, והיינו בהפסקה בכיתה, וישבנו ליד אחד השולחנות בכיתה, ואני זוכרת שכמה בנים בכיתה הציקו לי. אני לא זוכרת בדיוק למה הם הציקו לי, או בדיוק מה בדיוק היה שם, ואני זוכרת שהם כאילו היו מסביבי, אני ישבתי שם על איזה כיסא, או שהם צעקו בקול ממש רם, או שהם כאילו דיברו אליי לא יפה. הייתה לי תחושה מאוד מאוד לא נעימה, וזיהיתי ילד אחד כילד הדומיננטי, שאולי הוא היה הכי קרוב אליי, או שהוא הכי הציק לי, או... הוא היה הכי לא נעים, ובאיזשהו שלב פשוט באתי אליו ונשכתי אותו בצוואר. נשכתי אותו נשיכה גדולה, ואז...
Now, I'll give you another taste of this story, a few more seconds, but I want you when
Anat, I want to ask, I mean, some of the audience do not understand Hebrew.
Perfect, that's
so
great.
Maybe we have to explain to them what she was saying, or maybe not.
No, we actually don't. That's perfect that some of the audience doesn't understand Hebrew.
Oh.
Because what we want to look at as this different information channels and how they contribute to empathy. So some of you understood the story, and for those who understood the story, who understood Hebrew, it was very clear what she was talking about. For those who don't understand the language, you had to rely on visual cues, maybe on some paralinguistic cues, right? Listening to the intonation. And that's exactly what I wanted to do actually for a few more seconds to show you some of this video without sound. And try to see if you can get a gist of anything about, is she describing positive emotions, negative emotions? So let's do a bit of that. So I think you get the idea. So we had these participants come to the lab, tell their real autobiographical emotional stories. They were each asked to tell between 6 to 8 stories. They could choose how many. They could be both positive and negative, or only positive or only negative, to their choice. And we recorded physiological measures as they told the story. And then we showed them their stories again, their videos again. And we asked them using this rating dial, which you could see here, this appeared below their story. And they could move this line from negative to positive while they listened to the story. And we asked them to rate second by second what they felt. So this was a valence scale from very negative to very positive, what they felt while telling the story. So if this story by Shanit here was a story about what happened to her in kindergarten, not what she felt when she was in kindergarten and not what she feels now when she's looking at it, but to try to recall what she felt 10 minutes ago when she actually told the story. And then we have this second by second measure of what she at least thinks that she felt, believes she felt, reports that she felt second by second during the movie, right? We also said valence may not be enough. So I knew my colleagues from Stanford were recording these valence measures, but I felt that just having this negative to positive scale is not enough. Some of you understood what the target here was talking about. It was a very complex story, right? It may not be just negative, but have both sadness and fear and excitement and other emotions. So what we also did, and this wasn't done second by second, but after this 2-minute story, we asked each participant to rate during this story, how much did you feel happiness, sadness, fearful, excitement. We had 9 different emotions that they had to rate throughout the story. How much was this emotion prevalent? And now we have over 300 stories, both in Hebrew and in Arabic. So for a different stimuli set, we also brought in Arab students to tell stories in Hebrew and in Arabic. And we have more than 300 such stories where we have a video of someone telling a real story, their physiological measures while they told the story, how they rated their story second by second on this valence scale, and what specific emotions they reported feeling in each of these stories. And what we've been doing since is inviting people to the lab, or now we're doing versions of this task online, and having people watch these videos just like you did here. And we can ask them in some of our tasks to watch them in different conditions. This will be the focus of the rest of this talk. So to hear and see the targets telling the story, or just to see them, or just to hear them. We can also measure physiological measures from the observers watching the stories, and we can ask them to rate second by second what they think the target is feeling in each and every second of the story, and to then rate the specific emotions. How happy was the target, how sad, how excited, and so forth. And of course, we know what the target actually felt. So we can correlate these to measures. And that's why we can get a measure that we call empathic accuracy, right? It's not just a picture that you have to infer the emotion and then get, some wisdom of crowds, or what the experimenter asked the actor to portray. But we really know what the target felt each and every second. And we can compute this measure of accuracy. And this gives us a lot, there are of course a lot of, problems with this that we can talk about later on if you want, but it gives us a lot of opportunities to look at a lot of different things. So we can look at a correlation between these measures and look at how well you understand the other, given these different constraints, just the vision, just the auditory, etc. We can look at different clinical populations and say, okay, we know that these populations have some deficits in social interactions or in empathy, but now we can maybe pinpoint where are they better and when are they worse? What is their deficit really like? We can measure EEG or other measures, look at eye tracking measures and see what happens during these different tasks and so forth. So this is an example of how these results would look like for the continuous measure. You'd see here in blue what the target measure that they felt second by second during a video. You see it here, and you see the exact same line here. And you see 2 different observers, one that was really accurate, and if you calculate a correlation score, it will be really high, and one that was much less accurate. And this is an example of what we got from the specific emotion score, which was, I was really intrigued. I didn't know what we would find and how interesting it would be. It could have been that when we look at these results, we would just see that people more or less rate one emotion. I was really afraid, or I was really sad. And then maybe we would get enough information from the valence. But this is a typical result, both from targets and from what the observers report that the targets felt, or sometimes we ask what the observers themselves feel. And people really feel complex emotions and a wide range of emotions during these interactions. We now have tasks when we ask about a lot more emotions. I'll show you an example of this towards the end. And people in a 2-minute story say that they felt much more than just fear, right? In this 2-minute story, there was fear and embarrassment and pride and excitement and happiness and even to some degree angry and sad, right? So everything, the person felt everything. And this was actually really common to have all of these different emotions. And I think this shows us again, how complex real life interactions are and understanding other people is. So now finally to seeing some results. What do we know so far from these kinds of tasks? So one of the first studies that we ran, and this was a study run by Dr. Karine Yeshfe, postdoc at my lab, who also helped run all these, collect this information, collect the stimuli set from the targets. And the first thing we wanted to do is, like I showed you, to bring people, students to the lab and show them these videos in the full condition, audio plus video, or just the video, or just the audio. So everyone saw all conditions. Each person saw 9 videos, 3 under each condition, mixed in order and counterbalanced between participants. So each participant saw different videos in different conditions. They all saw the same 9 videos, but each participant saw them in different conditions. And we also had physiological measures, the same physiological measures recorded from them. I'll show you results from the heart rate information. So again, this is just describing the tasks. We recorded the targets telling their autobiographical events, and then we have their own self-report ratings. And then we showed these to observers who either saw just the video, heard just the audio, or heard and saw the video. And then we had their ratings too, and we could calculate a correlation between them. And this is the measure that we call empathic accuracy. And although we debated about this a lot during these few years in the lab, I think we all agreed that it's at least more cognitive, more related to the mentalizing aspects of empathy than to the emotional experience sharing ones. Or I'll say it differently, since there's still arguments about that. It definitely measures the cognitive measure, right? If you ask someone to report what the other person is feeling second by second, they can do it without feeling anything. You can do this task just as a cognitive measure. Some participants report feeling a lot. Maybe you felt it as well when you listened or saw this story. Some people really relate to that person and feel the emotions with them, but some do not. Some just, can report the feelings of the other without feeling too much with them. So the measure itself is a cognitive measure. We can also look at a correlation between the heart rate measures. So we have the heart rate of the target, and we have the continuous heart rate of the observers, and we can see how much they're in sync. And this kind of a synchronization may be a proxy of experience sharing, right? They're feeling the same emotions, or at least the same arousal measures. Is the task clear so far? Any questions?
Yeah, I have questions, but I'm wondering if
Here are the results. When you look at the behavioral measure, empathic accuracy, this is the correlation between this continuous measure. You see that, let's start with the video. When you just see the video, and I gave you on purpose these few seconds at the end, and some of you didn't understand Hebrew, which was great because you could see what you understood. And I think you understood some of it, right? You understand the gist. You understand that, for example, in the movie that I showed you, there's something negative going on. And people are really better than chance when they just have the visual information. But of course, they are much better when they have the semantic information. When they actually understand the story, they are much better than when they just see the video. And what was really interesting to us is that in the full condition, there's no advantage. You're just as good in the full and in the auditory condition. So if you just have visual cues, they give you something. But the visual cues don't give you anything above the auditory cues when you already have them. We see the exactly same results in these specific emotions. Here there was a complex computation. You can compute the difference between what the target reported and the specific emotions that the observer reported the target to have in different ways. But basically, to make it clear, we then normalized and made this in a scale from 0 to 100. And you were around here in chance level. So people, again, understand the specific emotions of others better than chance, even when they just have the video. But they're, of course, much better when they have the semantic information, or semantic plus the paralinguistic cues, right? When they have this auditory information. And when they have both, this doesn't add anything. So I think this may not be surprising, but this was in some ways surprising for us. And then lastly, when you look at the heart rate measures, there's actually more synchrony in the visual only condition. So in some ways, when all you have is these biological motions, when all you can do is this, if you want, this is an interpretation, this motor simulation, you get more synchrony with the other. But so if this is a more of a effective measure of experience sharing, it's a bit heightened in the visual only condition, but we still have to understand exactly what it means. And we might have some clues for this soon. And this is more than in the two other conditions. And if you have some thoughts about that, I'd love to hear them too.
Anat, I want to ask, do you want to take some questions now, or
So there are also some correlations, not strong one, and these correlations occurred mostly in the full condition. So when you have, and in real life, like I started saying, people measured these separately, but in real life, you both see the other and you hear them and you both try to understand them and you also feel with them. And all of these things happen together. And when we measured them in the full condition, when you had the full information, you also saw a correlation between the heart rate synchrony and empathic accuracy, and between the empathic accuracy, different empathic accuracy measures, the single emotions and the continuous scale. So this is the first full study that we have using these measures. Other things that we're now doing with them is we wanted to see what happens in EEG, and that's why I did want to focus a bit in my introduction on these, on this mu suppression measure. That's a proxy for mirroring, for stimulation, and we wanted to see whether this kind of suppression, this stimulation measure, is also related, associated with greater empathic accuracy. It's a study that I actually started doing with an American version of this task in Berkeley, and we're now trying to replicate. We had only 20 participants there, and we now replicated the results of a much larger sample of 60 participants in our Hebrew version here. So it's also a nice comparison between the tasks. And the task is exactly the same. So you're now familiar with this. We did exactly the same thing. We have this target self-reported ratings. We showed participants the clips in these different conditions. And this is a work with Desmond Ong, who was then at Stanford, now in Singapore, and my PhD student, Shir Genzel. And first of all, behaviorally, we see the exact same results. So these are, note, they're not exactly the same videos, otherwise it won't be interesting. We see them in the American version. We see them in our Israeli version here with a few videos that were changed. We learned some things from the physiological task. We wanted to use different videos in this task. So some of the stimuli was changed, and still we get these clear results where there's much more variance when you just have the visual information. That I think is obvious, and you saw it from your experience as well. You're better when you understand the story. That I think is obvious too, but there's no difference between the full and audio condition. So once you have the audio information, the video does not contribute to empathic accuracy. When we look at EEG, what we see is there's a correlation between mu suppression, this measure of simulation, experience sharing, if you like, and this cognitive measure of empathic accuracy. It's a negative correlation because I'm reminding you that mu is an index of mu suppression, right? Suppression in these rhythms is a proxy for neuronal activation. And when there's more suppression, there's also heightened accuracy across conditions here. What we're still asking ourselves, and this is part of the work that Shir Genzel is going to do in her PhD, is still what exactly does the sensory motor cortex contribute here to empathy? Is this simulation mirroring task? If it's more related to experience sharing, for example, one of the things that Shir wants to do now is not ask people what they think, not ask observers what they think the targets are feeling, but actually ask them what they themselves are feeling when they see these videos. And maybe when they report their own feelings, we'll have an even stronger correlation with mu suppression, because maybe what we're measuring here is their own experience. And we know it contributes just in some way to the cognitive measures as well. But this is really something that it's still under investigation. Questions about the EEG?
Yeah, Anat, can you hear me? Anat, can you
Maybe
hear
one
me?
thing I'll add here, for those of you who are familiar with EEG, it's important to note that they didn't do the task. It's a bit different because they didn't do the task inside the EEG. So inside the EEG, we just asked them to quietly sit still and watch the videos, because we didn't want the participants to do any motor actions and do these ratings while they watch the video inside the EEG, because that of course will elicit mu suppression. They're doing this motor rating, and we know that's what the measure measures most strongly. So we measured them silently when they weren't moving at all, and just asked them to focus on the videos. We also asked them some attention questions to see if they're really focused on the videos, and then they did the behavioral task outside the EEG and watched the videos again. And then lastly, Karin, the postdoc in my lab, Karin Yeshfe, is also doing this collaboration with Nachum Soroker in Bet Levinstein, where we really had this vision to not focus on specific lesions, right? Because there are lesions. I did work in my PhD focusing on frontal lesions and specifically on the IFG, which we know is related to mirroring. And there are other studies focusing on different regions. What we wanted to say is there's so many different regions that are related to mentalizing and to experience sharing. It really encompasses most of the cortex. Let's look at a large cohort of, we hope to have 60 different lesion participants, and then see if we can correlate different lesions to different deficits in empathic accuracy, again, showing these patients the task in the different conditions. Maybe some of them will be fine once they have the visual information, but lose it completely when they have just the visual. Some will will show deficits when they have to do some integration of both the visual and the auditory. Maybe some will have deficits in empathy altogether, and so forth. And of course, compare them to control groups. This Karin ran participants for 2 years, and it had to be completely halted for during COVID-19. So we now stop this completely and have to decide whether we're waiting to collect more data or analyzing what we have now, which is a bunch of very different lesions in very different places, both in right and left hemisphere and all around the place. What I can tell you already is that I looked at these 30 lesion patients that we have and their matched controls, which are all older adults, so ages 46 to 80. And behaviorally, we see exactly the same. So again, in terms of behavioral measures, we see that both groups, so also older adults, also lesion patients with different lesions, don't get any benefit from having visual information added to the auditory information. Once they have the auditory information, that's what they need for empathic accuracy. And I see that I have only a few more minutes, right, Sharon? Do we end at 1 or a bit later?
Do you hear me now? I'm not sure you do, Anat.
I couldn't hear you, sorry.
Yeah, I know, I tried to speak a few times. We're flexible. I mean, you have 5 minutes. If you need a few more, that's fine. Yeah. Okay.
I'm sorry, but did you want to ask questions?
We can. There are a few questions in the chat, and also we can have that left to the discussion at the end.
I want to show you one more thing that's really related to our last work that we're running now, and I'm really interested in hearing your thoughts. I'm sorry, because I turned my audio off to show you the video, I couldn't hear you guys.
No, it's okay. Happens.
So I think the summary is clear, right? We see in all these different behavioral tasks, we're still figuring out what we see in the brain or what we have in different lesions. But in terms of behavior, it's clear that video contributes something if you just have the video information channel. But once you have the audio, you don't need anything else for empathic accuracy, right? We see this here, we see this in all the other tasks. And this really relates to also very different lines of work. For example, Hillel Aviezer's work, some of you are familiar with this, who showed that people again and again, experiment after experiment, are sure that the important information is in the face and that they think they're focusing on the face to understand the emotion, but actually the emotion is much more captured in the context, right? In the body language, in the background, in the context, which in my case is in the linguistic semantic information. In Hillel's case is in other information that he gives the subjects. But there's the discrepancy here. People are sure they need the face, but actually they're not using the face. They're using other things to understand the emotions of others. And one of this question that we were left is why do we still feel that seeing the other is so important, especially for things like empathy and understanding others? And I actually want to stop and see if someone has some ideas, and then I can show you some initial data that we've collected.
I want to ask if when we actually hear the story without seeing the video, do we imagine, or you know, we imagine somebody, we imagine a face, or you know, a figure. And with respect, I also have a question about the auditory channel, but I'm now answering what you've asked.
So that's an excellent question. I don't know. We've never asked participants what they do. I'm not sure. I mean, when you talk to a friend on the phone, do you imagine them? Maybe you do. Maybe it's when it's a friend, you know, you do imagine the friend talking to you.
No, but you know how they look. I mean, think of, you hear the story, and if somebody you're not familiar with, do you have some, maybe even a vague visualization of the person? Let's say you hear, you can hear whether it's a man or a woman or a child, or you can hear somewhat their age. So how, like when you see biological motion from, you know, point light displays, you have some very vague, you animate it, or you to some extent. So I'm wondering if that also happens here in a way.
No, that's an excellent question. We have to ask our participants. I'll look into that. I don't know what people do when they just hear the other person. But any intuition from you guys? Why do we all feel, or most people feel, that they do need to see each other, and these results confuse them, because we feel that seeing the faces of others is somehow important in these social, emotional interactions?
It's comforting.
Okay.
Maybe we use the visual stimuli, for example, the face expression, as a kind of heuristic in real life interaction. But when you set a participant in the lab and you somehow force them to hear and be attentive to the auditory stimulus, then the auditory stimulus in of itself could be enough. But in real life interaction, we rely on visual stimuli as a heuristic, and that's why we feel it's important.
Okay, perfect. So you gave the two hypotheses that we thought about as well. So one of our own problems, I think, with the task that we've been using in the lab so far is we said, well, it's more naturalistic, it's more ecological, but it's still not really what happens in real life, because you're not really looking at the other person. So the target is not really present, and you're not really looking at them. And they may, you know, when they actually see the other looking at them, they may change their facial expressions. They may, and they may see when you don't understand something and then change what they say, and so forth. So maybe then the visual realm, the faces do contribute to accuracy. And second is what Sharon suggested. Maybe seeing the other person is really not important for empathy per se, or for empathic accuracy per se, but it's comforting, right? It contributes something to the more effective components of the interaction, to the feeling that you're listened to, that you're together with someone else. And since we weren't able to run lesion studies or EEG studies or eye tracking studies or dual EEG studies, or all the plans that we had for the semester in the lab, what Shia did is took advantage of the fact that we were all home under lockdown and on Zoom all day, and had 240 participants. This was done over the summer, and ran them on real live empathic accuracy tasks. So this is what she did. She had two people together each time on Zoom in pairs. One was told that they were going to be the storyteller and had to tell a short emotional story. And we wanted them now to be stories from the last few months. So not someone telling someone from kindergarten and someone telling something that was just happened. So something that a recent emotional story. And the other person was told that they were the listener. And after they told this, one told the story and one listened, they both had to rate what the storyteller had to rate what they felt while telling the story. So here there's no continuous rating. They finished telling the story and they just rate these specific emotions, but now we have 25 different emotions. And the listener rates what they think the storyteller felt, what emotions they think the storyteller felt. So this gives us this measure of empathic accuracy, right? And then they both answer questions about their affective experience, how much they felt they were together in this task, how much they had a sense that the perceiver, the listener, actually listened, how much did they have the sense that the perceiver was empathic, and so forth. And they did this in 2 different conditions. Half of the pairs, so 60 pairs, did this in the audio only condition. So they saw each other first. They said, this is going to be your partner. It's a real student at the university. They met each other, but now you're both going to turn your video off. And half the participants, the other 60 pairs, did this in a full condition, audio plus video, without seeing themselves. There's a way in Zoom where you can not see yourself. So this was just like seeing the other person. And what we saw here is that we looked first at this cognitive measure of empathic accuracy, right? To see if now we could replicate what we see in the lab, or maybe in this more realistic setting. It's still not completely realistic. They're not sitting together, but they do see each other. Maybe then seeing each other does contribute to empathic accuracy. And the answer is absolutely not. So again, we have these 2 scores. There's no difference between the groups. If something, the direction is the opposite, right? The audio here is a bit higher, they're a bit better, but this is not significant. So it doesn't, seeing the other does not contribute anything above hearing the other. But what about this confronting feeling that Sharon talked about, feeling listened to, feeling together, feeling that the other person was empathic towards you? Here we see big differences. So the sense of empathy, when we ask the perceiver, the listener, how much empathic did you feel towards the storyteller, they felt just the same. And note there's a ceiling effect, right? They all felt very empathic towards the other, but the storyteller felt very different. The storyteller felt that the other was much more empathic to them when they saw them. And the same for listening. So they felt that the other would listen to them much more when they saw them listening to them. And we're all familiar with this in the Zoom setting, right? Half of you are probably doing different things right now as I'm talking. We all have this feeling of at least we want to see the other person to know that they're there. And they both felt more together with the other when they listened to them. And by now, I'm not going to show you a replication study. We did this again with another 240 participants. We wanted to ask things that were a bit different, but basically we replicated all of these findings in another group of 240 participants. So really, it seems like in real life, social interactions involve different senses. We know that. We started with a focus on visual and auditory cues, but I want to remind you, one day we'll get out of this lockdown, we'll get out of the Zoom, and we'll get back to meeting people. And then we also have touch and smell and other things that affect our social interaction. But this was our first focus. And I showed you results of 4 different studies from our lab that indicate that hearing the other is most important for understanding them. And that seeing, if you just see, you understand something, but once you have the auditory channel, seeing does not contribute beyond hearing. However, seeing the other contributes to the affective experience. It contributes to being in some kind of physiological sync, to feeling together, to feeling listened to, and to feeling empathized with, although empathy might be the same, but you feel that the other person is much more empathic towards you. And I think this has implications both for basic affective science, right? We've all been studying for years what's there in the face and how do we recognize the emotions of others, but really we may be recognizing the emotions of others usually not by using facial expressions. So it goes back to implications for this basic affective science. Why do we still want to have to see the faces there? And it also has implications for our daily lives today in a global pandemic, when we talk to our grandparents, when we talk to our parents, when we talk to our colleagues, and so forth. So I think this is something to think about. And hopefully in the near future, we want to bring these more realistic studies to the lab using dual EEG to have people interact together. We want to continue studying clinical populations, our lesion patients, and the group of people with high functioning autism that we want to study both behaviorally and maybe also using these lab measures. And we want to start working on interventions for enhancing empathy. And with this, I really want to thank you and to leave room for your questions.
Wow, Anat, thank you very much. It was a beautiful talk. And I invite, first of all, everybody to give a big nod to Anat for a wonderful talk. Thank you very much. And I myself have a few questions, but maybe I'll open the stage for others to ask, and I'll complete that at the end. And there are also a few questions in the
I
chat.
can look through the chat if whatever you guys prefer, and you can also find me if I don't have time to answer everything. You can find my email, and I'd be happy to hear your thoughts and questions always.
Okay.
Okay, were there different groups of people who are especially good with visual cues, for example? So we haven't looked at specifics. So we wanted to, so far we see this overall, but of course there's a lot of variance. And I think when we go, especially to clinical populations, we'll go deeper into these differences. So when we think about clinical populations, such as people with different lesions or people on the autism spectrum, I think we will want to focus on these individual differences. It may be that we put people in this big group called autism spectrum, but we know that they're actually very, very different. And it might be that some of them actually benefit from having visual and auditory cues, and some of them would benefit from having just one cue and not the other, and will have deficits in integrating them. And someone asks about aphasia patients. So again, I think it could be that people with aphasia would benefit more from some of the cues. Yeah, exactly, from the visual cues. And we're now looking, so one of the interesting findings that we did see when we look at lesion patients now is when we compare left and right lesion patients, we see that some of the left patients do much worse on this task. And it makes sense after I told you this whole story, right? Because most of the information is in this linguistic world. But for most effective researchers, they think of the right hemisphere as being the important hemisphere for effective components. So, and there's a lot of data showing that right lesions show deficits in understanding emotions and understanding others. And really in this kind of task, it seems that the left hemisphere, maybe not surprisingly, plays an important role. Videos and subtitles, that's also a question I want to answer. We're looking into this now. So not only video and subtitles, but what happens if you just have the subtitles, right? What happens if you just have the linguistic information, but don't have these paralinguistic cues, don't have the prosody? So we're looking into this in different ways, just having the written text, having people from other languages hear the paralinguistic cues, but don't understand the language, and so forth.
I was also thinking along the same line. I just wanted to suggest, you have these mechanic kind of like robotic readers. They don't insert any effective information into the voice. So you do have some kind of like voice reading, but it's very, very flat. And I wondered if that, what would that convey? Because in a way that would leave the plot, I would say, but without the effective additional information envelope, the effective envelope on that.
Excellent. So we've been thinking about doing these for the last 3 years now. And I think the reason that we haven't really done any of these studies yet, and we're now starting, is that because we keep having these arguments of what's the right way to do it. So Sharon, the last thing you said seems like it's the cleanest manipulation, because you have the person speaking, but it's just in this, monotonic voice. To me, these always feel like they're very depressed people. So it's
or not people.
Or not people, right? But very depressed robots. So it's like we don't have, it's like this argument in effective science is that there is no neutral facial expression. And we have this tendency to, like we have a tendency to animate everything, we have this tendency to put emotion in everything. So even if you have this natural or neutral expression, we think this person is whatever, trying to hide something or upset. So I think also there isn't really a neutral voice. It will be difficult to do, but probably we should do all of these different combinations and then see what we get.
I see somebody wrote something in the chat. I also have a question before we go. I want to ask if you measured or have some index of the individuals who actually participated in how empathic they are? I mean, I'm not that familiar with this literature, but I'm wondering if there are questionnaires or some estimates, because you do see from your graphs, we can see that there is a lot of inter-individual variability in the way they rate the empathy, if it's along the videos, or if it, and I wonder if it also has to do, I mean, how correlated you are with the person who is telling, if it has to do with how empathic, what kind of how empathic you are as a person.
That's a great question, and it's something that we're actually struggling with. And there are empathy questionnaires, but the questionnaires are, of course, self-report questionnaires. One of the reasons we wanted to have this task is to make something that's different from self-report. On the other hand, we do want to see that it correlates to some degree with at least some of the self-reports. And indeed, in some of these studies, we find a correlation, small correlation with some of the scales. So for example, one of the empathy scales is one of the empathy questionnaires, one of the well-known ones is called the IRI, the Interpersonal Reactivity Index. And it has 4 subscales. One of them is perspective taking, which is this cognitive measure of empathy. And in some tasks, we see that it is correlated with our measures, but not in all of them. And I see the same with my colleagues in the United States. Sometimes in a big enough sample, you get these correlations, but in smaller lab samples, not always. You sometimes get a correlation with the AQ, the autism quotient. So people who are higher on this scale have more autism behaviors, and they do worse on these, usually worse on these empathic accuracy, real life paradigms. But these are small correlations and you don't always get them. I see there's a comment on using NLP and machine learning to try to compare this with human analysis. This is fascinating, and it's something that we're starting to do. So I have 2 computer science students now working on our Israeli data and also working with an American colleague on the American data and trying to do that. And one of the interesting questions that we keep going back to is, what's the truth? What do we want to compare these machine learning results to? Let's say we want to make these computers that can understand people better. Is understanding people, comparing them to the targets, to what the person said they felt, is that the truth? What's the ground truth? Or do we want to compare these machine learning algorithms to the wisdom of crowds, to what most people think the person felt? Some people, we can see it in our targets. Some targets, it feels like they have no idea what they felt. They just, they told the most frightening story, but they say they didn't feel anything. Did they really not feel anything? Who's right? Right? Should we have psychologists rate? And it goes back to clinical populations. I know we have to finish soon. I want to give you one preview to a beautiful study that a postdoc of now is starting now. I'm saying it's beautiful because it's hers. So I feel I can say it. It's really her idea and her work, Yonat, which I encourage everyone to follow. She's now in Cambridge working with Simon Baron-Cohen, and together with our lab, we're having this collaboration where we're going to film people, high-functioning people with autism, tell their own stories to see, and then rate what they felt, to see not only how bad or good they are at understanding others, but also how we are bad or good, how good are we at understanding them, right? We may, and that's a classic double empathy problem, right? We both have to we all have to understand each other. And we want to see, when do we understand, or when do we have deficits in understanding people from clinical populations? And then we go back to the question of who's right. If we say that the person felt sad, but they said that they felt happy or didn't feel anything at all, what's the truth? So I think it's the same when we think about these machine learning algorithms, when we think about clinical populations. We have to also ask ourselves, what are we trying to simulate? Are we trying to simulate what a psychologists would think is the truth, what the person said they felt, what their parents said they felt, what wisdom of parents would say, and so forth?
Anat, I want to ask, maybe the truth isn't important. Maybe it would be interesting to more or less cluster or classify groups of people. Maybe you could use, for example, I don't know, K-means or some clustering algorithms take all the people, and without any assumption on what the truth is, just see how, for example, people rated things, and see if that's transform them. I mean, if you get some clusters of individuals and maybe they have some, I don't know, similarities. I mean, I don't know. I'm just thinking that maybe the fact that two people felt something is similar, whether it is true or it isn't true, maybe that can characterize them in a way or reveal something about them.
Yes, definitely. Yes, I completely agree. So there's a lot of interesting questions to ask about the observers, what they think about the situation, and why different people feel different, think different things at different times when they see others. And then there's the question of what the target themselves say they felt, which is also, remember, one time point. So maybe when they were doing the task, they were tired or whatnot. And that's the data that we have. So we also have to take that into account. Someone asked if familiarity with a storyteller will have an effect. That's a great question. We don't know. What we did so far in the lab, because the first year we recorded these targets, most of them were students at the university. And then in the next year, when we showed the videos, we took out all the videos that observers said, I know this person, right? He's my TA, or he's my friend, and so forth. So we didn't analyze the data from the few people who knew the targets. And for the Zoom studies that Shir just ran, she asked only for people to be strangers in the experiment. So if they knew each other, they weren't paired. But it's an excellent question whether it's different once you see your child talking or your friend talking, and that's definitely something to look into. Thank you.
I also want to ask about the stories. I mean, people were telling stories that they felt had some, I guess, emotional component, or maybe even a big emotional component, because I'm assuming that otherwise they wouldn't have remembered that so much. And you can also see it in the rating where you show, okay, it seemed that each feeling or emotional type was rated to some extent. So it wasn't only happy or only sad or only pride. And I wanted to ask what would happen if people would be, I mean, and that's kind of like intense emotions, I would think. What would happen if you would parameterize in a way the emotional extent, or I would say ask somebody to tell some very emotionally boring experience. I don't know, maybe, I remember I had to walk to the grocery store, and then they were out of bread, so I got the other, something very boring. And what would then, maybe would that lower the synchronization? Would that, just a thought, you could parameters that to some extent, but still.
It's funny that you give that example, because we have stories like that. What happened in the Zoom study, because we asked people to give us recent events, and they were talking to someone else, a stranger, and sometimes they felt uncomfortable, and sometimes they didn't have very dramatic emotional events happening to them in the last few weeks. Some of them gave very emotional stories related to COVID-19 or whatnot, or just stress from exams and stuff like that. Some of them told stories like, I went to the bread store and they didn't have my bread, I was really frustrated, exactly like that. But we, of course, averaged all of them. So we don't have these differences. I think you're exactly right, especially in things like affective synchronization. I think we will have it. Like, I know Julia Goland, who is doing similar work at the IDC, to find really these physiological synchronizations, which for us is just, one study, but for her is really the focus. And we've been consulting with her a lot. To get these really strong effective synchronizations, you need really emotional stories. And she gets like the peaks of the peaks. If you're talking to a stranger and you really want to feel with them, she takes these very extreme emotional stories. And I think most of our stories are in between. They're not very extreme. And I think that's why a lot of the measures are weak.
I see. Maybe you should also, maybe you can analyze your data then,
split it,
classifying into some bins, or levels of emotional content or extent or magnitude. And
that's a great idea. Also, in the second study that I didn't have time to show you, in the replication study, what she also asked is she asked people to, the perceivers, to rate what they themselves felt, which is, so we don't have physiological measures, but it's also some measure of emotional synchrony, right? Experience sharing. So we can definitely, at least there, divide it into the more boring stories and the more emotional stories, and to see what happens there. Thank you.
so I don't know if there are any more questions, but are there? Well, Anat said she'd be happy to answer even directly. So if you don't have her email, you can email me and I'll email her, or I'll send you her email. Sorry, that would be even better. And so I'd like to thank you again, Anat, for a wonderful and beautiful talk. So thanks very, very much for joining us. And next week we have Tzvi Ganel with us, who will tell us about perception and perceptual interaction in 3D and 2D grasping. So welcome to join us in the same time. And again, thanks, Anat, for joining us.
Thank you for having me.
It was a pleasure.